A mature angiosperm seed is the final physical output of a strict mathematical algorithm. Inside that protective casing, the plant has packed two entirely separate genetic payloads, each engineered with a different chromosomal count. The first payload is the embryo. This is a diploid structure, carrying a paired set of chromosomes, and it is responsible for the genetic continuity of the next generation. The second payload is the endosperm. This is a triploid structure, holding three sets of chromosomes, and its sole function is to provide the nutritional support required to sustain the embryo. To engineer a single casing with these two distinct payloads, a plant must synchronize two separate pathways of cell division and execute a unique dual fusion event. The physical engine for this algorithm is the flower, which is organized into distinct reproductive whorls. The male whorl is the androecium. It relies on the anther, a bilobed structure where microsporangium develop into pollen sacs. The female whorl is the gynoecium. Deep inside its structure lies a hidden ovarian cavity that houses the ovules. Inside both of these locations, specialized cells known as mother cells are preparing to divide. These mother cells always begin in a diploid state. They contain a full paired set of chromosomes from the parent plant. The mathematical journey to the seed starts here, but the male and female mother cells will process their divisions with different efficiencies. This split-screen ledger tracks the biological accounting system required to produce usable haploid gametes from those diploid mother cells. On the left, the male system. The microspore mother cell undergoes one meiotic division to produce a tetrad of 4 functional pollen grains. Each of these mature pollen grains carries 2 distinct cells within a tough outer wall, a vegetative cell and a generative cell. On the right, the female megaspore mother cell also undergoes one meiotic division, initially producing 4 megaspores. But the female system operates under strict quality control. 3 of those megaspores immediately degenerate. The single surviving megaspore expands and develops into a highly specific female gametophyte, an 8-nucleate, 7-celled embryo sac. Compare the mathematical output. Male gamete production operates at a 1:4 ratio, while female production is restricted to a 1:1 functional ratio. This 1:1 functional ratio creates a biological scarcity. While pollen is produced in abundance for dispersal, the single embryo sac remains sequestered deep within the tissues of the ovary. Physically landing a pollen grain on the stigma of a flower does not guarantee the ledger will be completed. This line graph maps pollen-pistil interaction. This is a dynamic biochemical checkpoint, where the pistil tests the genetic viability of the pollen before allowing access to the ovary. In a compatible scenario, the pistil recognizes the correct pollen type. It promotes continuous, rapid growth of a pollen tube down through the style. However, if the pollen arrives from the same plant, the pistil identifies the genetic overlap and triggers a mechanism to arrest tube growth. This deliberate flatline is called self-incompatibility. By terminating self-pollination here, the plant guarantees outbreeding, introducing new genetic variables into the next generation. Consider this diagram of a mature embryo sac. For our model, we'll establish a baseline for the parent plant, a diploid state of exactly 24 chromosomes. Because meiosis reduces the chromosome count by half, we can deduce the haploid state. Any individual gamete entering this system must contain exactly 12 chromosomes. The pollen tube breaches the embryo sac through a synergid, releasing 2 distinct male gametes into the structure. In the first event, syngamy, one of these male gametes fuses with the single egg cell. The output of syngamy is a diploid zygote. 12 plus 12 restores the baseline paired count of 24 chromosomes. In the second event, called triple fusion, the remaining male gamete bypasses the egg entirely. Instead, it fuses with the 2 polar nuclei located in the central cell. The output of triple fusion is the primary endosperm nucleus. 12 plus 12 plus 12 achieves a triploid state of 36 chromosomes. This phenomenon is double fertilization. It simultaneously generates the diploid genetic blueprint and custom builds a triploid food supply to sustain it until germination. This equation workspace models a macroscopic problem based on our biological ledger. How much energy must a plant expend to produce a crop of exactly 200 viable seeds? To solve this, recall the requirements. Each seed needs exactly 1 egg cell and 1 pollen grain. On the female side, the constraint is tight. Because of the 1-to-1 functional ratio during megasporogenesis, generating 200 egg cells requires exactly 200 divisions of the megaspore mother cell. On the male side, the math is different. 1 microspore mother cell yields a tetrad of 4 functional pollen grains. So, producing 200 pollen grains requires only 50 divisions. Summing the requirements, the plant must execute exactly 250 meiotic divisions across its whorls to yield a target of 200 seeds. Plants optimize their mathematical energy expenditure. They front-load divisions to mass-produce cheap, disposable pollen, while strictly rationing the more demanding female divisions. Despite the chromosomal calculus of double fertilization, angiosperms have evolved clever mechanisms to completely bypass the ledger entirely. One anomaly is polyembryony, a condition frequently observed in citrus and mango crops, where a single seed yields multiple competing seedlings. The mechanism originates outside the gametes. Maternal sporophytic tissue, specifically the nucellus cells surrounding the embryo sac, spontaneously begins dividing. These rapidly dividing maternal cells forcefully protrude directly into the embryo sac, taking up space and developing into embryos themselves. This creates a ploidy loophole. Because the nucellus is already mature diploid tissue, it skips the requirement for meiosis and syngamy. Genetically, the resulting multiple embryos are genetically identical clones of the maternal parent plant. From the mathematical reduction of double fertilization to the clonal bypass of polyembryony, angiosperm reproduction follows a strict logic of genetic accounting.